Happy day, everyone. Today we are going to take a closer look at one of our major biological macromolecules, a macromolecule that we've talked about in previous units such as biochemistry, and we call this the structure of DNA. And you've probably heard about DNA, and if you're not sure, DNA stands for deoxyribonucleic acid. And deoxyribonucleic acid, along with the histone proteins, makes up our chromosomes. And these chromosomes are very important because they code for the proteins in our bodies. We will talk a little bit more about that process a little bit later on, but it's just important right now that you realize that proteins are what makes each organism its unique self. So the proteins determine what shape the cells will be in, what kinds of enzymes are going to be made, what kind of biological process will be there. And so proteins are a very important part of understanding organisms. Now, DNA has a very unique structure. It has a shape that we refer to as a double helix. And the double helix resembles a twisted ladder, similar to this little drawing here on the side. And it has some really unique properties that we'll begin to explore in some future sessions that help us with understanding how organisms work. So now we're going to look a little bit closer at the structure of our DNA molecule. The DNA molecule is made up of monomers, and those monomers are referred to as nucleotides. Now, a typical nucleotide is going to have three major parts. The parts are usually symbolized as a circle is the phosphate group, the second part is the pentagon-shaped structure, which is deoxyribose sugar. And the third part, which is symbolized as a rectangle, is going to be one of four nitrogenous bases. When we look at the structure of DNA and we take a look at the nucleotides and how they're arranged within the molecule of DNA, we can see that over on the sides of the ladder structure are the phosphate groups and the deoxyribose sugars. Then within the center of our DNA structure, which basically make up what we call the rungs, are the nucleotide bases, and those are arranged in pairs. In the center of the nucleotide bases are going to be hydrogen bonds. And those are important in the structure and the ability for DNA to easily replicate itself, as well as to be available for the transcription process. In DNA, there are two types of bases, the purines and the pyrimidines. The structure of DNA is designed so that there is one purine that matches up with one pyrimidine. Now, the purines are adenine and guanine, and the pyrimidines are cytosine and thymine. And one of the easiest ways to remember which ones are which is the pyrimidines all have a Y in their name, and the purines will not. So we can remember pyrimidines with cytosine and thymine, the purines adenine and guanine. In joining the bases together, we know that there's one purine that matches with one pyrimidine, but which ones go together? So one of the easiest ways I have to put them together is to remember that adenine always bonds with thymine, and cytosine always bonds with guanine. And how I remember this is I just kind of think of the way that I use the shortcut letter for these bases. So adenine is A and thymine is T. And if we look at the way I write the letters, A and T are both made out of straight lines. Whereas cytosine and guanine are both made with curved lines. So I remember that A and T go together and C and G. Now these also form different numbers of hydrogen bonds between them. As you remember from a previous slide, there is hydrogen bonds between the two bases. So A and T, there are 2 hydrogen bonds, so I always remember AT2, and C and G have 3 hydrogen bonds, so CG3. AT go together, 2 hydrogen bonds, C and G go together, 3 hydrogen bonds. Then I can always look at their name and I can see whether or not there's a Y present. The cytosine and thymine are my 2 pyrimidines, and the adenine and guanine are the 2 purines. 1953 is the year in which Watson and Crick discovered the structure of DNA. When they discovered this, they were not alone in their research methodologies. There was lots of other scientists that had contributing factors in the information that Watson and Crick were able to put together to figure out the double helix shape of DNA, as well as how the bases went together. Some X-ray crystallography was done in with the work of Rosalind Franklin and Morris Wilkins, and there was other people along the way that helped to contribute various pieces of information. And we need to think about this when we look at any scientific discovery. It's not just the one person or 2 people that we see the recognition go to. Many times there's a whole group of scientists that are working in the background, or they are building upon information from previous scientists. So that's an interesting side to take from this. But we're usually going to be talking about Watson and Crick when it comes to DNA, and now more and more we're seeing Wilkins and Franklin with their contributions more notably added in. So this is our section on the structure of DNA, and we'll move on to some more information in the next video. Thank you.